Hello, so this is my presentation. I wanted to make a recording of this just so you could see kind of how each slide would have been broken down if we lived in a perfect world. So this is my friend's daughter, Hermione, and his wife, Christina. So Hermione, she has cystic fibrosis, right? And he and I were talking, and one of the things that's kind of different, like I've never heard of this before, and I think that you really wouldn't know about it unless you were part of like the CF cystic fibrosis community. Is like they like hot sauce a lot. Like a lot of people that have cystic fibrosis seem to really enjoy spice, and I was wondering if maybe there's a genetic reason for that, if that had, if eating spice helped the symptoms of CF or something. Is there a reason for why it's so common in the community? So that is kind of what I started out looking for. Okay, so a little bit about cystic fibrosis. I feel like it's one of those diseases that you kind of learn in every intro to genetics class because it's such a, it's not a very common disease, but it happens enough and it's got such a clear distinction for why it occurs. So there's a single mutation on the CFTR. This is typically a gene that codes for a protein that creates chlorine channels. So you can put chlorine in and out of cells. When it's mutated, there ends up being a bunch of mucus that can build up in the lungs. And I didn't know about this before, but it's actually you have a lot of people with CF have a lot of digestive issues. This gene is located on chromosome 7, and then this is what I was talking about when I was referencing the genetic classes. It's an autosomal recessive disorder, so both parents have to be a carrier, and there is a 25% chance that their children will have cystic fibrosis. This is just a chart showing where CFTR expression is found. So in the lung, when you hear about cystic fibrosis, I feel like everybody kind of thinks lungs initially, which, yeah, there is expression there, and it is the same as your glands for like your spit. But really, like there's so much other expression that's in the digestive areas. Of all of that it's doing for the lungs, obviously, yes, if you cannot breathe, that's going to be the most important thing. But the digestion issues that they're having as well, there's got to be something there. So this is a slide that breaks down the different types of mutations. So F508 is the mutation that I am looking at throughout the rest of this presentation. It's the most common one. It varies based on what you're looking at about how common this one is. See here it says 66.8. This one says 70% of people with CF have this type of mutation, and later on you will see another study said it was 80 something. So, but general takeaway, this is the most common mutation that occurs with the CFTR gene. This is a little bit breakdown of how that mutation occurs. So this is typical. This is a healthy CFTR. And then over here, it gets stopped at this point. So it gets misfolded and it never gets to continue on. Yeah, this is where it's saying 88%. So you don't know, No, we don't know. This is what happens. So typically this is going to code for phenylalanine. In the F508 deletion, it's missing this part. And so phenylalanine is not able to be produced, right? It's not going to be able to be coded for, and it ends up with the misfolding. When it is not misfolded, this is what it looks like. This is from AlphaFold. And then taking a step back from cystic fibrosis for a second and focusing a little bit more on the taste and spice. So reason I'm not saying spice is taste is because it's actually technically not. It's more accurate to say you're feeling spice than it is to say you're tasting it. But I still wanted to get genes that are associated with all of them so I can see if any of them are connected to CF through the different programs later on. So a couple little things to note. TRPV1 is going to be the type of receptor that detects capsaicin, which is in chili peppers, which is in a lot of hot sauce. And then ASIC3 is just kind of a generic response to spice. So it detects pH changes. It's not going to detect a specific spice, just kind of like a generic general. Keeping an eye on the different types of channels that we have going on here, since cystic fibrosis is a disease that's caused because of a channel misfunction. I was just trying to just keep track of these, but they end up not really being that important. What is important is this bitter right here, the TAS2R, and I add quite a bit more genes later on that are related to this. You'll see it ties in a lot with future studies that we will talk about. So the next step that I took was to go to Reactome, and I entered the genes that I had found. So I got their unique product session number, entered them into Reactome, and just wanted to kind of see what I would get starting out. So this is one of the first results that I got from Reactome. This is pretty cool. It does end up linking all of the genes that had to deal with taste together, as well as spice. So interestingly, like, we were able to associate taste and spice, but not, anything with CF, which, to be honest, I wasn't really expecting anything with CF, but I wanted to include it just in case. This is the CFTR F508 mutation. So typically, CFTR is a primary ABC transporter, which means it works with the rest of its family to pump chlorine in and out, right? Then over here, we can see what's going on when it's mutated with the F508 mutation. So every single time it gets misfolded, it just gets sent to be degraded. It's never able to interact with any of the rest of its family. Up here, I just wanted to include this real quick. This is one of the medicines, Olfactory. This is one of the 3 most helpful medicines for people that have cystic fibrosis. And actually, it's usually included, trifecta is it's included with the other 2, and typically it's a very successful treatment for people with CF. So this was my original conclusion, was that I couldn't figure out a way to link the 2. So ultimately, I did not stop there. We still have plenty of slides to go, but I originally started to think that maybe there was a physical component to spice breaking down mucus and making it easier for people to breathe. Maybe something like that was going on, and it wasn't actually genetic, it was more physical. But we have some more to talk about, so let's go to it. So using GWAS, we gathered more information, right? So typing cystic fibrosis in, we can get to different studies, different associations, and find a whole bunch more genes associated with cystic fibrosis, which is very important, right? So the next thing that we did, thank you, by the way. So we got tons of genes for cystic fibrosis. These are genes that are going to be talked about later from a specific study. These are the bitter genes. Then we have a couple genes. We have spice, we've got salt, sour, just a couple, just to see if anything pops up, And then over here, this is one of the features of Reactome. You can see the F508 mutation. These are other types of mutation for cystic fibrosis, but we can see our little F508 right here with a cluster of a couple other ones on chromosome 7. That's part of locus zoom. Just like I said, it's available on GWAS. So using the larger list of genes with Reactome, we were able to get quite a bit more information this time. So you can see sensory perception is overrepresented, and we were able to get just a couple more flowcharts for, like pathways. So this is cool, right? But the main thing that we actually wanted to look at that was brought up to me is overrepresentation in the platelet homeostasis. Okay, so what does that mean, and what does it have to do with CF? We will get there. But first, we need to look at it a little bit. So this is one of the diagrams that's included with that study. So now that we know that platelet homeostasis and taste and CF can all kind of be related, might all be related, we can go to Google Scholar and look for an article that can help. So targeting an olfactory receptor mitigates high residual platelet reactivity and arterial thrombosis through actin cytoskeleton depolymerization. Oh my God. What is this study about? I don't know, but I need to. So the first step, understand what the study is, why they did their study, and if/how it relates to us. So the very first step, I like to do this with any kind of title that's long. Just break it down into easier parts. It's gonna make it just so much simpler and honestly quicker to understand the whole point of the article. So the article, what it's about, is targeting odor receptors to lower high amounts of leftover reactive platelets and lessen the formation of new blood clots by breaking down actin found in the cell's structure. So what this study is, claiming that they did was saying, like, they're saying that they were able to target odor receptors to improve cardiovascular disease, which is kind of a wild idea. Like, the odor receptors are supposed to be in, you know, a very specific region of our brain that detects odors. How is this a thing? And this spoiler alert kind of thing, they're not just there, they're throughout our whole body, these receptors, and they were able to target them, help lower blood pressure and different cardiovascular things, which is interesting. But how does this relate to cystic fibrosis and taste? So in this paper where they were looking at olfactory receptors for new treatments for cardiovascular, they quote this article, and you can see it cited at the end there, but it's ORs are known to be expressed ectopically in non-olfactory tissues such as airway, smooth muscle, kidney, colon, etc. So what it's saying is that ORs are not just where we'd expect to find them in the brain, but they're expressed elsewhere as well. So what we did, looking at like the articles that first stated that, 11, 13, 12, 11 is the one that I thought was the most interesting, right, for cystic fibrosis. So what is this article about and how does it relate? The article's title is Bitter Taste Receptors on Airway Smooth Muscle Bronchodilate by Localized Calcium Signaling and Reverse Obstruction. So airway smooth muscle is going to be ASM from now on. What does this mean? So bitter taste receptors, again, T2R38, remember from when we had first gathered that little collection of genes, is a bitter taste receptor expressed in the sinonasal tract and elsewhere. Having both of these is related to having a better quality of life in cystic fibrosis patients. What I mean by having both of these is having both alleles. ASM bronchodilation is the process of widening the airways in the lungs to improve airflow and breathe easier. Obviously, this is going to be helpful for people that have CF if their airways are clogged with mucus. It's not getting rid of the mucus, but it's giving them a little bit more space to have air. Speaking of having more space for air within the lungs, reverse obstruction is just the process of, you know, opening up the airway a little bit. It's showing great promise as a form of treatment for obstructive lung diseases. So that's what this study is kind of getting at. So what are the results, right? That's what we want to know. They're interesting. So the first thing that we want to look at is this chart over here on the side. So these are all of the taste receptors that they had looked at. This is the expression of the taste receptors in ASM cells. So you can see not all of them are there, but some of them are there and some of them are there a lot. Now, ADORA, the 3 at the end, ADORA 1, ADR, those ones, those are used as just kind of controls to, you know, get a baseline for evaluating the other expression. Figure C is showing that the bitter tastings perform similar to other well-known bronchoconstrictive agonists. So basically just saying that the response to calcium was similar to that of other bronchoconstrictives, agonists that are typically found in medications and stuff that we currently have on the market. It's, you know, trying to say that it's promising, right? All right, so figure 2.A shows the dose-dependent relaxation of isolated intact mouse airways utilizing the bitter taste receptor agonists chloroquine, ditonium, and quinine. The beta agonist isopropanol was also used and recorded for reference. So isopropanol is a typical bronchodilator. It's not used really in inhalers, but it is used to help open up the airway. And so they wanted to use that as a baseline to kind of see if the bitterants, how they act in comparison to it. And if you can see here, I mean, they act faster. They act much stronger and much faster than isopropanol. So the type of bronchodilator typically prescribed to CF patients are known as SABAs, short-acting beta agonists. These act on the beta 2 receptors and were not included in that last figure that we just looked at. So instead of isopropanol, it would be one of these. So actually, the albuterol is used as the control in the procedure that follows as part of this study. So what is the study? What did they do? Basically, they sedated mice and they induced an asthma attack with methacholine. I think that's how you say that. Not entirely sure. Methacholine is a bronchoconstrictor, so like I said, it's simulating an asthma attack. And then what they did is used inhalation types of the bitter tastants. So, and the positive control, albuterol. So they gave some of them the typical medication that you would give a CF patient, albuterol, and they gave them what they're studying, the bitter tastants. And then, let's see, this is what they found. So, this is actually really good. This is the baseline. This is just a mouse that was under anesthesia. These two are the mice that were given asthma attacks, but then given nothing for them. And then over here is the two that are kind of the big takeaways. So this is the bitter tastant, and this is albuterol, the inhaler form, like the current treatment. You can see here, this is how much the muscle relaxed. So the current medication that we're using, it only relaxed it this much. It's 4 times the amount. So it's showing that the bitter tasting actually worked a lot better than the current available treatment on the market. And then that's just kind of repeating what I just said. So guanine worked better than albuterol, right? That's the takeaway from the study. But this is showing an association between bitter taste and pulmonary mechanisms. And then lastly, it's showing a way in which pulmonary diseases such as CF, asthma, these types of things can potentially be related to taste. So these are just some quotes directly from the article from the study. So again, reiterating that bitter tastants relax the airways in a mouse model much better than the current form of treatment. So this kind of leads to our new conclusion. We are still unable to find a single genetic explanation for why some individuals with CF really like spicy food, but we were able to show that OR expression is present in airways, meaning that bitter tastants may bind to these receptors to produce a relaxation effect. I believe it is possible to suggest that consuming bitter foods may provide a level of relief to those with CF. Since bronchoconstriction is such a common issue among those with CF, and since bitter tastants provided faster relief when compared to the current CF bronchoconstriction treatment. I think my primary future consideration, if I was to keep going down this avenue and looking at these things, is to see if there's an dopaminergic effect that occurs when bronchoconstriction is relaxed. Because specifically, if you're eating food that has lots of bitter tastants, if you're inhaling any of this stuff and it is giving you a relaxation effect, is that effect strong enough to have changes to your personal preference? Is that enough to get you to say, I really like the way I feel after I eat this food. I really like this food. That's kind of my thought process there. But some other considerations for the future. So we have some research is available that CF patients really like turmeric. Some studies show that different species are unable to feel pain related to various spices. For example, capsaicin, which is like we talked about it earlier. It's the main ingredient in chili peppers. Not ingredient, but it's how we feel the spice from it. The evolutionary basis for why we have bronchodilating bitter taste receptors on ASM is still debated. We don't know. So there's different pathways there you could look into with evolution and different species and turmeric. These are the citations that I have. There's a couple. That's it. Thank you.